GCSE Biology. Welcome. Are you excited for a fun learning session? Make sure you have your book and a pen before we start. Today you will learn about enzyme models. Enzymes action. Enzymes are large 3D molecules made by a protein folding into a specific shape. The shape that a protein folds into is dependent upon the order of the amino acids that are present in the protein. The active site of the enzyme is the site where the substrate binds. The lock and key model suggests that enzymes have active sites that are specific and will only bind to particular substrates. When the substrate binds to the active site of the enzyme, an enzyme-substrate complex is formed. During synthesis, the two, or more, molecules bind to the active site of the enzyme. The enzyme catalyzes the reaction and the two molecules are bound together. The enzyme must then release the new molecule. This frees the enzyme to find another two substrates to catalyze. In digestion, the molecule binds to the active site. The enzyme catalyzes the reaction and the product is released as two or more molecules. When the enzyme releases the new molecule, this frees the enzyme to find another two substrates to catalyze. Enzymes have optimal, ideal, conditions under which they have the fastest rate of reaction. There are conditions that can affect the rate of the reaction of an enzyme. Enzymes have specific pH that they work best at. For example, the proteases in the stomach work at pH 2. Enzymes in the human body work best at around 37 degrees. If they are exposed to temperatures that are much higher or lower, they do not work as well. If an enzyme is exposed to extreme temperatures of pH for too long, then the bonds in the protein are broken. If the bonds break, the shape of the active site changes. If the shape of the active site is altered, then the substrate will no longer be able to bind with it. The enzyme is said to have denatured.